Hi, it's Dr. Robert Sykert. Thanks for tuning in to a new episode of the Doctor Podcast Show, and I'm your host and creator of Doctor Podcast. My voice is a little raspy today because I had a bad cold last week, but I'm getting over it, fortunately. If you like the show, which I'm sure you will, this is a really exciting episode. Please click on the subscribe and like button and repost button, and we appreciate that because we'll get more and better guests. And speaking of better guests, today we have an incredible guest, Dr. Mark Hamilton. Dr. Hamilton is a hematologist, oncologist, and cell molecular biology researcher at Stanford University in California, one of the top medical centers in the USA and the world. And he recently published an extremely important article in the New England Journal of Medicine, which is the top medical journal in the world as well. And we'll talk about that as well. So, Mark, thanks very much for taking time to come on the show today. Really appreciate it. So tell us how you got involved in this specialty and how many years of training have you had since the usual 4 years of medical school?
So after postgraduate, I did an MD and a PhD, and so those together were about 10 years. And now I'm completing around the 6th year of my postdoctoral education as a fellow here at Stanford. So it has been a very long path. At Stanford, I've been very fortunate to be involved in the lymphoma and cell therapy programs, working with Dr. David Miklos and Ash Elizadeh in their laboratories. The thing that I love about hematologic malignancy is I really love inpatient medicine. It's something that I've loved since I was a medical student, and especially working with oncology patients. But the thing I really enjoyed was you can take a patient with a hematologic malignancy, give them, when they're very sick, extremely powerful therapies, be it induction chemotherapy or CAR T cell therapies that will inherently make them more ill over the coming weeks. But at the end of it, they can end up cured on the other end of the therapy. And I just thought that that was the most incredible thing that I had seen in medicine. And it has such a powerful impact on patient lives while also allowing for a very broad medical practice. And that's how I ended up where I am.
It's an incredible story. So you've been in training basically for about 16 years after medical school, is that right?
16 years after my undergraduate school, but it
Oh,
has been a long road.
wow. So I recently interviewed some neurosurgeons and cardiac surgeons. They only did 10 and a half years. So you're way ahead of them, but they said they were pretty close to retirement after all those years. But it sounds like you're just super energetic. So you recently published a major article in the New England Journal, as I mentioned, dealing with the CAR T cell therapy, which stands for chimeric antigen receptor T lymphocyte therapy. So we'll call it CAR T for short. Tell us about that, what that is, and how it's a major breakthrough in treating the hematologic malignancies such as lymphoma, especially.
Yeah, absolutely. So CAR T cell therapy, and especially in lymphoma, has kind of been the dream realization of cellular therapies. And in this therapy, we're able to take T lymphocytes out of patients' bodies and treat them with a virus that basically educates them in how to kill the tumor in a relatively specific way. Once they express this CAR T cell receptor, we can put them back into the tumor, and the CAR T cell will expand over the course of approximately a couple of weeks, and it will functionally destroy the tumor in that period of time, we hope, and put the patient into a complete response. One of the powers of the CAR T cell therapy is that it's a living therapy, and so it does stick around after it initially expands, which may have some value in continuing to monitor for cancer relapse and making sure that the cancer stays dead once it is killed.
Right. That's really fascinating. So just to back up for people in the audience who are not physicians, as you know, there are red cells and white blood cells, which make up most of the cells in your blood. The red cells carry oxygen. The white blood cells are the cells that protect us from infections, viruses, bacteria, fungus, and they also are supposed to protect us from getting cancer. and specifically the lymphocytes, the B cells and the T cells. The B cells produce antibodies, which protect us from infections. But tell us what the T cell lymphocyte does in the body to protect us from cancer.
That's exactly right. And so when I usually describe to patients what our different white blood cells do, I kind of describe it like an army. And when I think of an army, you need lots of different divisions and types of units. And so if you think about a neutrophil, which is the primary way that we kind of run the front lines of our immune response, I think of those kind of as the infantry, and they go in and attack bacteria right at the source. And as you mentioned, the B lymphocytes, I think of as the artillery. They are able to kind of shoot immune globulins out, which seek and destroy bacterial cells, viral cells, or anything else that they've kind of been previously exposed to. And when I think of the T lymphocytes, they're kind of a mixed bag, but I tend to think of them more as kind of generals or specialists, some of which can work directly as assassins or assault units, and some of which can kind of direct other immune cells. And so you can imagine this would be a very powerful arm of the immune system to be honed in directly on the cancer. And so when we think of the CAR T cells working, this is a cell type that's really able to go directly to the tumor and mediate its destruction, but also recruit other cells to facilitate that destruction too.
So they're like the Green Berets or the Rangers or the Navy SEALs of our immune system, basically.
I think I use the Green Beret one the most often, but
Yeah,
all of
that's
those
what,
work.
right. So if that's the case, then why do people get these hematologic cancers, especially lymphoma or leukemia? Are the T lymphocytes asleep, or are the cancer cells somehow tricking them and camouflaging themselves so that the T cells don't recognize them?
That's a great question. And, when I think of this, I actually try to give some credit to the cell that the cancer used to be. The cancer really wants to be a normal cell, but it's developed some degree of genetic disruption that makes it abnormal and specifically abnormally proliferative. And the immune system is very good at tracking this and understanding when it's happening and destroying it right in its infancy. But because it's your own cells, and the immune system knows to be tolerant to your own cells, it can sometimes be very difficult to do that. And these tumors can, with enough genetic disruption, with enough growth, escape the immune system. And many times, if we look in a tumor, we will still find that the immune system is active in it, but just unable to kind of keep up with the pace of the tumor.
So the tumor somehow evades the normal immune system and then grows and then kind of overwhelms the T cells that are supposed to attack it, basically.
That's
So,
exactly right. And the
right,
other cells that also are doing their part too.
right. And that's what a lymphoma cancer basically is, right?
That's precisely right. So a lymphoma cancer is a cancer of the lymphatic system, which is going to be B cells and T cells. And B cell lymphomas are by far the most common. When we talk about them, we think about Hodgkin lymphoma and non-Hodgkin lymphoma. And CAR T cells are really a therapy that's most effective in non-Hodgkin lymphomas. And the most common of those are going to be large cell lymphomas, where CAR T cells probably do most of their work. And then things like mantle cell lymphomas, and marginal zone lymphomas, where CAR T cells are also functional. On
Right.
the other side of that are leukemias, and then the major leukemia that we use CAR T cells in is B cell acute lymphoblastic leukemias, both in children and adults. And they do function in those tumors as well, but are less likely to be curative. And then there's additional tumors that and leukemia types that the CAR T cells are currently being approved for, such as chronic leukemias.
I see. So tell us what the process is. Well, how were these patients treated before the CAR T cell therapy came, and when did it come, and how did we treat these patients before, and what was the success rate with chemotherapy or radiation?
Yeah. So I'll take the primary case, which will be large cell lymphomas, where CAR T cells have really made the largest splash in the last couple of years, since there's so many different kinds of tumors that these are used in now. Large cell lymphomas are very aggressive lymphomas of the immune system that can fortunately be cured, but as you can imagine, they will require aggressive chemotherapies in any line that they're treated in. And so when we think of chemotherapies, we think of which line we're treating, with the first line being the very first treatment. And we always want to put our best foot forward. This is what I tell patients. We want to use our best therapies first. And in large cell lymphoma, that's usually a therapy called R-CHOP, which is a combination chemotherapy that's really relatively well tolerated and can cure between 40 to 60% of patients in the first line, depending on the type of tumor that they have, the type of large cell lymphoma that they have. And before CAR T cell therapies, the next step, if somebody were to have relapse disease, would typically be doing an autologous cellular transplant that involves high dose chemotherapy, and then giving patients basically their own cells back. And this was reasonably effective, but many patients would relapse, or because they had just been treated with the chemotherapy, wouldn't respond to the chemotherapies that they were given. And so this was an inadequate treatment. CAR T cell therapies were initially approved around 2017 for large cell lymphoma in a study called the ZUMA-1 study for a CAR T cell called axi-cell, and several other CAR T cells, one called Tisacel and one called Lysacel, were also approved around that time. And then subsequently, CAR T cells went up against autologous cell transplant in additional studies, main one being the ZUMA-7 study, and were found to be superior to autologous transplant.
The autologous transplant is like a stem cell bone marrow transplant, basically?
It's a stem cell transplant. That's exactly right. But it's a transplant of your own cells, and the cells are usually collected peripherally.
And they're collected and make sure they're the normal cells, not the cancerous cells.
That's exactly right. And one important point is that large cell lymphomas typically won't circulate. And so because of that, we wouldn't be collecting them.
I see. All right. So tell us how CAR T cell therapy is done. So you have a patient and they've not had a good response, unfortunately, to chemotherapy, and the stem cell transplant. What do you do to their cells, and how do you do it to make them now the Navy SEALs or Green Berets more effective?
So when we take a patient, especially a patient with large cell lymphoma, one, there's a time limit on what we're doing. The clock is ticking because the cancers can grow very quickly. And so when we get referred these patients after, now usually it's in the second line, so after they've had R-CHOP and they've been refractory to that or relapsed shortly after, then we would plan to treat somebody with CAR T cell therapy. And the first thing we would do is evaluate them to see how much disease they have. If they have too much disease, we would consider doing something like a bridging therapy, which is using another form of therapy to control the disease. So the first thing is always making sure that the patient is going to be able to get the therapy and that they're going to be safe while waiting for it. The next step is going to be apheresis. We basically, Dr. Miklos describes this as Jiffy Lube. We take patient blood out of their body. We harvest the white blood cells out of it. And then we put the blood back in, and we do that for a number of hours until we have enough peripheral white blood cells. And then the next step is sending it.
And this is done by a machine that is able to filter out the white cells that you need?
That is exactly right.
So it's done, you put an IV in the arm, you take the blood out, the machine separates out the white cells from all the other blood products, and then you have a bag filled with the cells that you want.
That's exactly right. And so that's something that we do at the home institution, and there's kind of apheresis chairs, and these machines are useful for a number of different things. But in this case, it's harvesting the white blood cells out of the patients. And then those cells are sent to a genetically modified product facility. And typically that'll be, in almost all cases, a commercial facility. And so there's a number of commercial entities for which CAR T cells are approved, and they will produce the CAR T cell usually along the lines of about 3 to 4 weeks from when we send them the blood. Here at Stanford, we have numbers of clinical trials where we do make our own CAR T cells, and that's common among kind of major centers throughout the world. One of the big ones that is about to be published in the Lancet from us is the CAR-22 study, led by my colleague, friend, and mentor, Dr. Frank, as well as Dr. Miklos. And that's just another example of a CAR T cell coming out of kind of the academic realm. But the majority of these are produced by companies.
So
In the
these
case,
are pharmaceutical companies or biotech companies?
yes, these are pharmaceutical companies that are kind of generating the product. And it is an expensive therapy, which is a major critique, costing about $500,000 per bag. And this is a limitation to our ability to give patients CAR T cells.
Wow, $500,000 per treatment.
$500,000 for the bag.
Is it covered by insurance or Medicare?
This would typically be covered by insurances, of
It
course.
would.
And so we have to
So that-
be careful to stay within the approvals.
Right. So now what do these companies do to the cells that you sent them in a bag from the patient who has the cancer?
So the cell will take the white blood cells, generally through kind of proprietary processes, but basically we take the T cells themselves. They are infected with a virus, and this can either be a retrovirus or a lentivirus, with the important part of this being that the virus will integrate DNA into the cells. So this is a genetically modified product and will have kind of man-made DNA built into it. And that DNA is going to code for the CAR T cell receptor, with the kind of canonical receptor being FMC63, which is an antibody directed against a CD19 protein. And CD19 is just an antigen expressed on B cells and B cell cancers specifically. There are any number of antigens that can be targeted by a chimeric antigen receptor. The BCMA is another kind of famous one used in multiple myeloma. But that's really the business end of the CAR T cell, is the targeting receptor. The majority of ones that are currently in commercial use are going to be targeting CD19. And the rest of the structure is a little bit different in terms of the different products. So when we describe these, we usually describe them as kind of being the same type of CAR, but with different engines. And so when we think of a CD28 engine, we've got an entire metaphor base around.
Right. It's easier to understand.
When we think
Patients.
about like a CD28 engine, we think of that like a Tesla. It's very, very, very fast. The CAR T cell is going to ramp up very quickly, and then it's going to kind of slow down more quickly after that. It's going to lose its charge. Whereas if we think of a 41BB type of vector, which is just another kind of signaling domain that we use, that would be like a Prius. It ramps up kind of a little bit more slowly. It'll take a little bit more time. You're less likely to go off the guardrails from the speed, but it's going to last a little bit longer. It's going to kind of get you all the way from San Francisco to LA. And, you know, both of those cars are going to take you where you want to go. But one or the other may be right for the right patient.
Right. So let me back up a little bit. So they are using, these labs or biotech companies are using a virus where they've emptied out the virus's DNA. They put in this new manufactured DNA into the virus. The virus then bathes the cells that the patient gave, and the virus actually invades those cells, the T lymphocytes, and injects this new man-made DNA into the T lymphocytes. Am I right so far?
That's exactly right.
And then this DNA codes for RNA, which then codes for protein, and the protein attaches itself to the surface of the patient's cells. Is that right?
That's right. The protein will go to the surface of the T cell, and that protein will help the T cell recognize kind of the cognate antigen. It's kind of like a key to a lock.
So they fit in together. The new receptor on the T cell attaches to the receptors on the cancer cell that are floating around the patient's body.
That's exactly right. And so the major receptor would be CD19, but this could really work with anything expressed on the surface of a cell.
That's pretty amazing. So the company then sends back this bag of lymphocytes that have been basically changed with new man-made DNA and protein. And then the patient is given that intravenously. Is that how it's done?
That's exactly right. So the patients will come in, they'll undergo first something called lymphodepleting chemotherapy. This tends to be about 3 days of chemotherapy. And the goal of that is really to kind of clear out the white blood cells so that the CAR T cells have room to expand. On the fifth day after that, which we call day 0, we'll infuse the CAR T cells in what is usually a fairly boring procedure. And then the CAR T cells will expand slowly over the course of the next week. And that's
When
really
you
where
say they expand, they start multiplying and growing inside the patient's body.
that's exactly right. The CAR T cells, they don't stay still. They recognize these antigens and it causes them to grow exponentially. And that exponential growth is what leads to you having enough CAR T cells to ultimately overwhelm the cancer.
So these new cells that have this new protein receptor smell out the cancer cells and then attach to them like a lock and key, and they start destroying the cancer cells.
That's exactly right. And
That's
hopefully by the end, there's no more cancer. And
right. And the reason this is called a living therapy, as you mentioned in the beginning, is these cells basically hang around and stay around. Do they stay forever, or how long do they stay in the person's body?
that's a great question, and it's one that we study quite closely, actually. There is a publication out of Dr. June at Penn's lab describing kind of the longest CAR T cell that we know of, hanging out for about 10 years, which is to
10
say
years.
about as long as we've been giving these therapies. The longest that I have seen them personally in my own research is 4 years, which is as long as we here at Stanford have been giving them. So they can persist for a very long time. That's not to say that they do in every patient. Um, and it's important for us to tell patients that at some point the cancer might be gone, but the CAR T cell might still be there. And that could lead to a toxicity.
Right. I was just going to ask you that. Do these new cells with this new surface protein ever attack the patient's other cells or organs mistakenly, almost causing a autoimmune-like reaction?
Yeah, so that's a great question. And we have what we call on target but off tumor targeting. And so the important thing about an antigen like CD19 is, like I was saying, the cancer really still wants to be, in some ways, a normal cell. And that is an antigen expressed in normal cells, specifically in normal B cells. And so the CAR T cell is not going to only kill the cancer itself, but it's going to kill anything that it sees that expresses that molecule. And so in the case of CD19 directed CAR T cell therapy, there is a destruction of B cells leading to a B cell aplasia that can persist for a very long time post-treatment. And that is an important toxicity because B cells, like I said, were an important portion of the immune system.
Right.
Um, and you can survive without them. Uh, and we do hope that they'll come back eventually as the CAR T cells slowly die off over time. But it is a risk to the therapy.
I see. What about, I think I read about cytokine storms where there's a huge reaction to these new cells and lots of toxic chemicals are released. Can that happen?
So that's a great question. And when we think of the CAR T cells, I usually tell patients that there's kind of 5 toxicities that we need to worry about. 3 of them are a little bit earlier and 2 of them are a little bit later. So the first toxicity is what you're talking about, which is cytokine release syndrome, which can also be colloquially called a cytokine storm, but cytokine release syndrome is the technical word we use, at least here at Stanford. And we describe those as flu-like symptoms. But just like the flu, this can also be very severe and even deadly. And this will be true for all of the toxicities with CAR T cells. The next one we describe is immune effector cell associated neurotoxicity syndrome, or ICANS. And that's functionally confusion, but confusion can also lead to brain swelling and be dangerous if not treated correctly. The next after that is going to be something called an immune effector cell associated hemophagocytic syndrome, or IECHS. This is a newer entity, but it's basically an out of control immune response in the bone marrow, leading to profound cytopenias. Uh, After
All
that,
the bone marrow cells are kind of being destroyed.
that's exactly right, from an inflammatory response. And we do think that, there may be inflammation in the bone marrow that occurs from this therapy. Leading from there, there's cytopenia and infection. This is actually the most deadly toxicity of CAR T cells. So all of these toxicities that I'm mentioning, when we look back at all of our data, we can treat them effectively during the immediate course of the treatment. And we expect them to happen about in the first month, and then they're kind of gone, they die off, and for the most part, they aren't going to come back. But patients are at risk for infection for a very long time after CAR T cell therapy, and it continues to be a danger even years out. And so that's something that we have to really caution patients against, is that if patients are getting an infection, and especially during the era of COVID, if they got COVID, we really wanted to know about it because they were at high risk for this. And then the last major toxicity we talk about is second primary malignancy or secondary tumors.
And
And
that's
this is
what
really
your
what
paper was
our
about in the New
paper is
England
about.
Journal, right?
That's exactly right. And so that's a newer toxicity. And so all I have to say is there can be other toxicities, especially in some of the other CAR T cells. There may be additional toxicities that come up over time as we have more and more CAR T cells coming online. But those are the major ones for the CAR T cells that we typically use in the lymphoma clinic.
Tell us about the publication in the New England Journal of Medicine that you had dealing with whether there's a increased risk of getting cancers after the CAR T cell therapy.
So an important background to this is because, lentiviral and retroviral vectors integrate into the DNA of the patient, there's always a concern that it could disrupt an important portion of the DNA and cause a cancer. Basically, the integration itself could cause a cancer, and this would, theoretically be a T cell cancer. And this was realized kind of back in the early 2000s in a group of young boys receiving cell therapy for SCID treatment, where they actually did get secondary T cell lymphomas from their gene therapies. And of course, this would be a very bad side effect because you could give a patient a secondary cancer derived from your product. And then beyond that, there's always a risk in many treatments, many cancer treatments of other malignancies. And these are often things like myeloid malignancies, such as myelodysplastic syndrome. And those would be commonly associated with chemotherapies and then also stem cell transplants. And so around February, the Food and Drug Administration put out an announcement regarding CAR T cells, stating a risk for second primary malignancy, and in particular, myeloid and T cell malignancies, with a concern that some of the T cell malignancies may have viral vector integration into them. So this kind of dreaded concern of secondary vector-containing CAR T cell malignancies. And they put out quite a large announcement asking for any molecular biology studies surrounding these cancers, should there be reports of them. And we've been studying cytopenia in CAR T cells for quite a long time. And we had happened to have a second T cell malignancy that we knew about, but also we needed to make sure that all of our patients were safe. And so we instituted basically a system-wide, taking all of our patients, search for any of these secondary lymphoma or secondary malignancies. And we got as many as, we believe we got all of them, and to try to understand what the actual risk was. And so in part one of our study, we took about 724 patients. This was 791 CAR T cell infusions because some patients get more than one CAR T cell. And we said just how many of these patients are getting malignancies, especially hematologic malignancies. And that number was about 6.5% at 3 years. And that's about what's been reported in the literature. It's going to vary a little bit. And those numbers are relatively low. So I believe that that was about 14 total patients having either a myeloid or lymphoid malignancy. And that would be similar to our other treatments, such as an autologous stem cell transplant. And just one of those patients had a T cell malignancy. And the major question became, was this derived from the CAR T cell vector, from a CAR T cell? And if not, how did it come to be? Because if we can kind of understand how these are coming about, then it can either reassure us or make us more concerned based on that answer.
So did you find there was an increased risk or not really significant?
So that's the right question.
Right.
First, when we kind of looked at this, this is just one case. And our case stood out, we think, because of the depth of the analysis that we went into. We had very broad profiling of many of this patient's samples, including longitudinal plasma samples, longitudinal tumor samples. We profiled both bulk DNA, which is just kind of all the DNA meshed together, but we also did an analysis of the single cell resolution using kind of more advanced technologies that let us really isolate the cancer and say, is there something specifically in this cancer versus all other cells? And, so the first thing that we saw in that was there was not CAR T cell vector in this specific cancer. There was none. We showed it at the protein level, at the DNA level 3 times, and at the RNA level. And so it was a very clear illustration of how these analyses could go about and different ways that you could try to detect this, which is one of the kind of important things about it is not that, since it was just one patient, it was that's a relatively small number of patients clearly, but
Right. That's 1 out of 724. But what about, you mentioned that 5 or 6% had other malignancies, but they're not related. Can you explain that a little more?
yeah. So when we say 6.5% at 3 years, that means after 3 years, 6.5% of our patients ended up with a hematologic malignancy, specifically, and the majority of those were myeloid, so MDS or AML. And what's important about that is that those are kind of expected malignancies to pop up in patients who have had chemotherapies and cellular therapies. And
Right.
so possible that CAR T cell therapies can contribute to these malignancies, and we're studying that further right now. But it's also possible that those would have come about from the chemotherapies or other therapies these patients had been exposed to.
Right. So it seems to be pretty safe. And we need to keep in mind that these patients had no other choice. This was a last resort because their previous treatments, the standard type of treatments, had failed. So if they didn't undergo the CAR T cell therapy, they would have unfortunately died. Is that right?
That's right. And that's an extremely important point, is that uh, in these patients, when we think about the risk and the benefit, when we're talking about a cancer where we're worried about a few weeks of waiting to give them a treatment, the risk of kind of the distant risk of them maybe having a secondary malignancy is worth the benefit of killing this immediate, very aggressive cancer. And I don't think that anybody really doubts that now as these studies are coming out. These second malignancies are just not occurring at such a high rate that they would stop us from treating these already aggressive cancers. In the case of the T cell malignancy, there is a risk of T cell malignancy in patients who have these B cell lymphomas anyways. And in our hands, it was about the same as the reported literature. That is to say that we saw these malignancies at about the rate we would expect just from patients that weren't getting CAR T cell therapies that had B cell malignancies.
Right.
And so it was very, very low.
Now, these virus vectors that you're using to inject this new DNA to produce the protein receptors, for example, the doctor I spoke to recently about injecting genes under the retina, they were using an adenovirus-associated vector. You're using different viruses here. Does each biotech company have its own specific virus that they use as the medium for injecting the new DNA?
So each company does have a slightly different version of the virus, but some of the major viruses used are going to be the lentiviral and retroviral systems. There are other strategies, and so I think you mentioned CRISPR of gene editing, and other ones exist, including TALENs. And so these can be very useful ways of modifying cells beyond just putting in the vector. And so we expect that additional viruses or additional genomic modifications are going to kind of come out in the future that
Right.
make CAR T cells new and possibly more effective, those would have to be individually analyzed for their risk of these secondary malignancies. And that's undergoing strict review processes right now.
Right. I don't know if you saw the article came out in Nature yesterday about a new gene editing procedure. I think it's called bridge editing, where they can actually edit single nucleotides in DNA. It sounds really incredible. It sounds like it's the next generation of CRISPR, but much better. And it sounds like that will be eventually applied to this as well to make it more effective and safer. One other question I had in your paper in the New England Journal, mentioned that some of the patients were found to have Epstein-Barr virus floating around in their system. Epstein-Barr virus, very common virus, causes mononucleosis or mono, which a lot of people get. Most people have no problems with it, but there is an association with Epstein-Barr virus and certain types of lymphoma. Can you tell us the significance of that?
Yeah, I think that that's the other half of kind of what we did with this patient is after we excluded the possibility that the CAR T cell might have caused this cancer directly through vector integration, we next looked to see why she might have gotten this cancer from other means. And so Epstein-Barr virus is a common virus in children, as you stated, but it's also a common cause of lymphoma in patients that are immune suppressed. It typically will have post-transplant lymphoproliferative disorders will often be Epstein-Barr virus driven, and they occur in patients that have had transplants in the past and are typically on immune suppression. In this patient's case, her large cell lymphoma, the B cell lymphoma, was EBV positive, and she had had a history of immune suppression too, likely explaining why she got the original lymphoma. And then importantly, her T cell lymphoma was also EBV positive. And so in her case, she had likely a very profound underlying susceptibility to these virally driven cancers. And this was demonstrated also in the fact that she had mutations in certain genes, including TET2 and DNMT3A, that were present in both tumors, suggesting that they were kind of in her stem cells, unfortunately.
Do we know why some people just get mononucleosis and they're fine after Epstein-Barr, and other people it seems to trigger cancers? I think Burkitt's lymphoma is one of them, if I'm not wrong, and maybe nasopharyngeal carcinomas. Is that right?
That's exactly right. There's a number of different cancers that can be driven by the Epstein-Barr virus. And in the case of Burkitt lymphoma, it can be different between kind of children who get this kind of canonically in Africa and adults who would get it in the United States. For most of these EBV-driven tumors, immune suppression plays a role in it driving the EBV reactivating and driving the tumor. There's some kind of critical component of the immune system that's lost. And the way that the Epstein-Barr virus works is that it just can cause proliferation of lymphocytes very effectively. And then, in other cases, likely just bad luck. I should add to this one thing, an important point to get across that was missed during the conversation is that there was a companion article in the New England Journal on the same day as us that also presented a second T cell lymphoma. And I think that, it's important to put this, put it into that context, because that lymphoma was CAR T cell positive. And there are scattered reports of other CAR T cell positive lymphomas. And so I do think it's important to also, mention that ours isn't the only story out there, though it's an important contribution. There may be instances, or there will be instances, we're sure of it, of these vectors contributing to T cell lymphomas. But all of that said, it's still extremely rare, and the therapies are still very, very safe. And then one last point is, the aggressive lymphoma point's a very important one, that it's definitely the risk balances the benefit in these cases. If CAR T cell therapies move to more benign conditions, such as autoimmunity, it may change the risk benefit scenario a little bit. And so right now on the front lines of cancer, I think that a lot of what we're doing is trying to inform the safety of CAR T cells in conditions that aren't as aggressive as cancer and try to understand what those risks may be in the future, especially because the population treated is going to be healthier and also much larger. So I just wanted to add in those 2 points before we ended.
Right. Important point. So you've been at Stanford now for how many years?
I think I'm entering my 6th year right
Sixth
now.
year. And you're finishing up on this hematology, oncology, and research fellowship in the future, right?
That's exactly right. So I'm coming kind of to the end of training, which I'm very excited about.
Right. Do you know what you're going to be doing in the future yet?
I have some prospects that I am really excited about and looking forward to, but I don't know where exactly I'll be yet, be it here or somewhere else. But
That's
I
great.
gotta say, I'm very fortunate to have been able to work here in any capacity for my
Yeah.
training.
What's it like working at Stanford, which is, as I mentioned, one of the world's top medical centers, innovative research, great doctors? What's it been like working there for so many years?
The thing that I found very powerful about Stanford, other than just, the remarkable support we get, the very broad and wonderful patient population that we have that trust their care to us, has really been the team science approach that we take. And so, I'm doing this interview on behalf of my co-authors, Takeshi Sugio and Troy Nordenboss, as well as the PIs on the study, Dr. Elizadeh and Dr. Miklos. And, the importance of this is that we all work together to achieve common goals, and we all have different skill sets that we can apply to a problem. And everybody is willing to work together in this context, and it's made for some very strong research efforts out of our lymphoma and cell therapy group, and we kind of all continue to work in that way. And so I think that that's kind of the main secret sauce here, just beyond it being a fantastic research institute. And I'm very lucky to be a part of that effort.
Teamwork with great people. Nothing's better than that.
Nothing's better than that.
So you're doing incredible work. Your research is helping many people and will continue to do so. I want to thank you, and I'm sure the public watching this want to thank you as well, and hope you continue to do this incredible work, which is very meaningful to thousands of patients who have these conditions. So thanks very much. I also want to thank you for taking the time to talk with us today and explain these complicated treatments. I understand it better. I'm sure the audience does as well. So I want to thank you very much for your time.
Well, we're thankful for the opportunity and for the time that you're taking to help disseminate this important information.
All right. Thank you, and take care. Stay well.